In this session, we're going to cover the absorption of nutrients across the GI tract. We'll talk initially about absorption across the small intestine. So most of absorption processes that occur are in the small intestine itself, and about 90% of absorption occurs there. And so it's involved in the absorption of your monosaccharides, such as glucose and fructose, your amino acids, your fatty acids and monoglyceride, which are obviously a breakdown product of triglycerides, your electrolytes, such as sodium and potassium, your vitamins, and your water. The absorption of calcium and iron, they're adjusted to the body's needs, so you take what you need. For example, calcium absorption is regulated through vitamin D levels, but the rest of what you take into your mouth and eat is fully absorbed. So therefore, the more you eat, the more is absorbed. And that's because your GI tract releases the appropriate amount of food and the appropriate amount of enzymes to fully digest and fully absorb at any one point. So you get slow release of food from the stomach into the small intestine, so it can be fully digested and absorbed, for example. Most absorption occurs in the duodenum and the jejunum. Remembering, of course, these are the initial parts of the small intestine. So food gets churned around in your stomach and then is slowly released as chyme into the duodenum. And then the duodenum receives pancreatic juices and it receives your bile salts and your bicarbonate, where it allows it to start that digestion of the nutrients to occur. So there's some digestion's already occurred, of course, but most of it occurs in the small intestine. So most food stuffs or products of food digestion are absorbed in that duodenum and the jejunum. A much smaller amount is absorbed in the ileum and the terminal part of the ileum, which is obviously called the terminal ileum, before the food waste moves into the colon and you get final absorption of things like water. There's a lot of bacteria in the colon as well, of course. There are specialized transport mechanisms for vitamin B12 that we've already discussed, because vitamin B12 is absorbed in the terminal ileum, and actually vitamin B12 binds to intrinsic factor within the stomach, because intrinsic factor is released into the stomach, and that helps to prevent its degradation within the stomach itself. And as you will already know that intrinsic factor levels are vitally important for vitamin B12 absorption, and things like pernicious anemia, where you don't have intrinsic factor being released, mean that you can become vitamin B12 deficient. Bile salts are also reabsorbed in the ileum, and that's so that they can be reused for fat absorption. And in total, about 9 litres per day are typically absorbed. So we have a little look at the structure of the small intestine itself, and in particular the villi and the microvilli. We have this nice villi, which is shown here, and on the surface of this you can see these little microvilli, these little hair-like structures. And you have about somewhere between 3,000 and 6,000 of those microvilli per epithelial cells per cell, and they serve to increase that surface area, therefore increasing surface area for absorption, but also for where you can have enzymes present. Because of course we have those enzymes that are present on the microvilli, such as your lactase, your maltase, your sucrase, your peptidases, which end up doing the final digestion of your dipeptides, for example, and your disaccharides. And this increased surface area also allows for nutrient absorption into the intestinal capillary network. So here you can see the capillary network in this villi. And that capillary network drains into the hepatic portal system, which directs the blood to the liver for nutrient processing. So remember, the nutrients that you take in through your diet get absorbed through these villi and microvilli into your hepatic portal system, then go straight to the liver, where the liver takes the nutrients that it needs, but also helps to process unwanted materials that may be getting through and into that hepatic portal system. We're looking a little bit more at the structure. You can see the lamina propria there. That's just connective tissue. Remember the enzymes, these are membrane bound, and these are associated with the microvilli. The cells themselves, these epithelial cells, they are joined by what are called tight junctions, and that means it limits very well luminal contents between cells, so stops stuff kind of squeezing in between. And of course we have nerve fibers there, which can detect pain, for example. So you have pain receptors there, as an example. We also have the lacteals, so that's shown in this little green tube, if you like, and they're basically modified lymphatic vessels, and they're present for fat absorption. So because the fats are too big to cross the epithelial cells and pass into the capillary network, they pass into to the lacteal network instead. And of course, what follows the fats are your fat-soluble vitamins. Now, we all know how absorption across the small intestine can occur. It can be an active or passive process, or a combination of both. We can go through passive diffusion, just going from high concentration to low concentration. And in this case, of course, lipid-soluble molecules cross much easier. You have facilitated diffusion, so where there are specialized proteins that allow the transport of other molecules, water-soluble ones, for example, to allow them to cross, again, from high concentration to low concentration. So it's passive, it doesn't require any energy. And finally, you may have active transport, where you're using energy in the form of ATP to power against a concentration gradient. So in this case, going from low concentration to high concentration. So therefore, you need energy to go against that natural diffusion gradient. So when we're thinking about absorption, the first one we'll cover is carbohydrate absorption, specifically looking at glucose itself. So two monosaccharides, so glucose and galactose, they use what is called the SGLT symporter. And so if we're looking at the SGLT symporter, the first thing to remember is the levels of sodium. Now you might think, why are you thinking about sodium when we're actually focusing on glucose and galactose transport? Well, it's because it's a symporter. So sodium, through its gradient, drags glucose into the cells, into the epithelial cells. So what we have at the basolateral membrane, so that's in linking up with the interstitium, so it's in contact with the interstitial fluid, you have a sodium-potassium pump. And the sodium is pumped out of the cell, and the potassium is pumped into the cell. And what this means is we have a low concentration of sodium within these epithelial cells. So then if we go to the tubular lumen, so this is our GI tract, sodium then diffuses through the SGLT symporter down its concentration gradient in the cell, and that by the same token drags glucose in with it. So what's being used is the energy of the sodium concentration gradient dragging glucose into the cell. So no ATP is used at this particular point. So to get glucose into the cell, we're not using ATP, but of course, in reality, we are using ATP to generate the sodium gradient because we have this sodium-potassium pump that is working, that is using ATP. So glucose enters we now know that glucose enters the blood passively by facilitated diffusion using GLUT2. So remembering what's happening, glucose comes in through the SGLT1 symporter into the epithelial cells, and then down its concentration gradient through the GLUT2 transporter and into the bloodstream. So that again is facilitated diffusion, but it's going down a concentration gradient. Now, there are similar mechanisms for other carbohydrates as well, but this is just a nice example to show you how glucose is actually transported across the epithelial cells using one type of transporter, and then from the epithelial cells into the blood down a different type of transporter. We're now going to go into fat absorption. So, just a little reminder from an earlier session, your bile salts, which are made in your liver, stored in your gallbladder and then released when food enters the duodenum. These act as detergents, and they help to convert your large fat globules into a lipid emulsion. So you get all this mechanical digestion, which breaks up your fat, and then your little drops of fat get emulsified by the bile salts. This then allows, remember, the lipase to act on them, and you end up with a monoglycerides, your fatty acids, but also you have cholesterol and fat-soluble vitamins in there. And what you end up with after this degradation through lipase is what are called micelles. And in these micelles, your lipid core contains all of these things. So your monoglycerides and fatty acids, because of the degradation by lipase, and your cholesterol and your fats and your fat-soluble vitamins. These micelles, like I've said, contain your triglycerides, which have been broken down, your cholesterol and your fat-soluble vitamins. So these components of your micelles, they're obviously lipid soluble because of what's in there. So they can diffuse through the plasma membrane of cells. So here are your monoglycerides, here are your fatty acids, and they just pass by diffusion from the GI tract lumen through the epithelial plasma membrane and into the cytoplasm of those cells themselves. Once they're inside the cells, they get resynthesized. The monoglycerides and the fatty acids are resynthesized into triglycerides, and then they coalesce into droplets and are coated with a layer of lipoprotein. And they then, in this form, they are what's called chylomicrons, and they're quite large. So they then pass by a different process. So they're not just simple transport like your glucoses and your amino acids. They have to pass through a process called exocytosis. And because they are much larger, they then need to pass into the lymphatics, not the blood. And they enter into the lacteals. Now, the chylomicrons are very important. They help make the fat droplets water soluble. So basically, the fat droplets are inside, and the outside of the chylomicron is lipoprotein, and that's water soluble. And as I said, the chylomicrons, they do not enter the blood, and that's because they can't cross the basement membrane because they are just too big. And the lacteal doesn't have such a membrane, hence why it can pass into the lacteals and then into the lymphatic system. So as we've just shown here, your chylomicrons, they're water soluble because of this lipoprotein. They've got a hydrophobic core because it's got triglycerides, cholesterol, etc. within them, and they go on to enter the lymphatics rather than the capillaries themselves. So we now know that chylomicrons have entered the lymphatic system, and they're returned to the venous system near the heart through the thoracic duct. So if we're looking at a summary as to what's happening at this point, we're in our intestinal lumen. We have a large fat globules are there, and you get all this mechanical digestion through contraction and relaxation of your smooth muscle, and through the action of your bile salts, you get emulsification of your droplets. They become much smaller, increasing the surface area. The bile salts and the lipase are then acting on them to break down the triglycerides into monoglyceride and fatty acids, and you form these micelles, which again are nice and small. They then just diffuse, because they're lipid soluble, into the intestinal epithelial cells, where they're converted, where the monoglycerides and fatty acids are reconverted to triglycerides, and you start to form these chylomicrons, where you've got a fatty hydrophobic core with a hydrophilic surface, which is made up of your lipoproteins. They enter the lacteals and the lymphatic system, and then enter the blood near the heart. So the chylomicrons are basically delivering their lipid components, then they've entered the blood, they deliver their lipid components to the adipose tissue, to cardiac tissue, to skeletal muscle for use and for storage. And excess chylomicrons are taken up by the liver and packaged into your VLDL particles and your LDL particles. So that's your very low density lipoprotein and your low density lipoprotein. The VLDLs, they'll leave the liver, they'll enter the blood, and they'll be converted to LDLs. And obviously, we know that they have a high cholesterol content. HDL, as you may well know, is made in the liver, and that removes excess cholesterol from LDLs. Hence why HDL is good, because it takes cholesterol away from the system, whereas LDL is bad at high levels. But you do need it because it's your cholesterol carrier around the body. So like we said, your VLDLs deliver triglycerides to skeletal muscle and adipose tissue, whilst LDL delivers cholesterol to peripheral tissues for your steroid hormone synthesis. So remember, steroid hormones are made from cholesterol, examples being vitamin D, aldosterone, cortisol, testosterone, estrogen. These are all made from cholesterol, so therefore you need cholesterol receptors in the tissues. So you have them in the testes, for example. For testosterone, you have them in the ovaries for estrogen and progesterone, for example. You have them in the adrenal cortex for cortisol and aldosterone, for example. Excess LDL, as we know, can lead to the buildup of LDL plaques in blood vessels, leading to atherosclerosis. But HDL picks up excess cholesterol in the blood and takes it to the liver for removal in bile. And if we're looking just at a size of each of these particles, you can see that the chylomicrons are very, very large, in particular compared to VLDL. IDL is intermediate density lipoproteins, so in between LDLs and VLDLs. And then you've got your high density lipoproteins there as well. And of course, a high HDL to LDL ratio reduces your risk of a heart attack because you've got lots of good cholesterol and lower amounts of bad cholesterol in terms of ratios. So here are a couple of review questions for you. Press pause and have a read, and then we'll go through it. Which of the following are chylomicrons composed of? So proteins, that's correct. You've got your lipoproteins, that's what helps to make up the chylomicrons. Cholesterol, yep, that is also correct. You have cholesterol within the core of the chylomicrons. Triglyceride, that is also correct. That is within the core of the chylomicrons. Fat-soluble vitamins, that is also correct. They are within the core. And your water-soluble vitamins, that is incorrect. Your water-soluble vitamins can be transported around the blood, dissolved within the blood itself, because they are water-soluble, as is your plasma. Which of the following are correct regarding absorption of nutrients? Most water is passively absorbed in the small intestine by osmosis as other nutrients are taken up. That is correct. Number 2, the water-soluble vitamins are easily absorbed along with water. That is correct. Quite logical, really. Vitamin B12 is bound to intrinsic factor and is absorbed in the duodenum. That's incorrect. Vitamin B12 is, of course, absorbed in the terminal ileum. Fat-soluble vitamins are absorbed as part of micelle constituents. That is correct. They are fat-soluble. And the fat-soluble vitamins are A, D, E, and K. That is also correct. Now, an extra little question for you. What are the key features or functions of vitamins A, D, E, and K?